Okay, gene drives and you, but probably not you. Probably something else. Let's talk about gene drives and mosquitoes specifically. So, mosquitoes have males and females, yeah, as you would imagine, and yeah, it takes a male and a female to make little baby mosquitoes. I would say before we go through this, normally if we're in class, I would try to take everybody's temperature. How do we feel about mosquitoes? I mean, you got the one side where you would say these are things that, yeah, they're alive, they're here on the planet, even right here in Texarkana. They maybe have as much right to be here as I do. And then there's the other side of things that say, boy, I really don't like all these itchy bites and the potential to spread diseases, and maybe I wish there weren't so many mosquitoes. With technology like a gene drive, we can have dramatic effects on a mosquito population, and the risk is with a gene drive, you can actually drive mosquitoes to extinction. You can drive a particular species completely off the face of the earth. And I don't know, that's a big responsibility. And the thing is, I don't believe there is a governing board that gets to decide who eliminates what species or doesn't. Because the technology is widely available. And basically, by going through this class, you're learning at least the basics of how to operate the technology. And so it's fairly accessible as well. Frankly, there's a lot of YouTube videos out there. Yeah, a lot of which are actually really good. I'm not going to link to all of them, but if my explanation is not doing it for you, oh my gosh, can you get a second opinion? Yeah, always seek a second opinion. There you go. So, the way mosquitoes work is they actually have three chromosomes. Two of them are going to be autosomes. The third one is going to be a chromosome that's more like the X chromosome, but it's going to come in two variants. And we're going to call those variants the X and Y variant of the third chromosome, chromosome 3. So X and Y contain almost all the same genes, but there's a particular gene on there where they have either the male or female specific version. So, yeah, their X and Y chromosomes are still pretty much identical, whereas in humans, the human and actually most mammalian Y chromosomes actually a broken off arm of an ancient X chromosome, we think. And so this is very old, and there's really not much left that actually codes for protein on a Y chromosome in a human or another mammal. Anyway, the gene on the chromosome 3 that determines whether a young mosquito is going to grow up to be genetically male or genetically female is called the double sex gene. And yeah, there's the female version of the double sex gene, and if that's turned on, it will repress all the other genes that would make the mosquito turn into a male. And it will activate all the genes that turn the mosquito into a female. So you've actually got to activate a whole bunch of different genes, because you think about it, yeah, there's a lot of parts of males and females that are different. I mean, every single cell is different, for starters, but also larger body parts end up different. And so it takes a lot of different genes. You can't just, turn on one thing and expect everything to be different. This is kind of like the master switch. So I guess you do turn this one gene on, but it works by operating lots of other genes. So there may be hundreds of other genes that it will actually affect. The double sex male gene is just like a double sex female gene, except that it's got a little genetic difference, and it will have the opposite effect. It is going to activate all the genes for male differentiation, repress all the ones for female differentiation, and you're going to end up with a male phenotype. So remember, phenotype is what a thing looks like, or what effect the genes have had. The genotype is what the DNA says. So within a mosquito, actually within every male human, there are all the genes that you would need to make a female human. Within a female human, you're actually missing one gene that you would need to make a male human, and that's a gene that's going to be carried on the Y chromosome. So that's the big difference there. But yeah, this is the main difference. All right, how does the double sex gene work? And this is going to be part of your test answer. You're going to be telling me about the double sex gene and how it is going to how a gene drive targeting the double sex gene could drive a mosquito species to extinction. So, The difference between the two forms of double-sex gene, and up here in the corner, I don't know if you know these little symbols. This little one with the arrow pointing up at an angle, that's going to be male. So this is supposed to be like Mars, the god of war, and that's supposed to be his shield and his sword, or his spear, rather. Because, yeah, all right, ancient humans were sexist pricks, I guess. Down here at the bottom, we've got the symbol for females, which is meant to be a mirror. Because, yeah, once again, the ancient people sexism thing. The idea was this is a symbol for Venus, but the idea was that since she's female, she's always going to be looking at herself in the mirror and preening. Yeah, that's fairly vile, but that's where these symptoms, yeah, they are kind of symptoms. These symbols come from. The difference between the genes is subtle. So, in each of the genes, there are going to be several exons which will be expressed. In the double-sex male, they are going to be missing just a little sequence right here that would make this chunk right there into exon number 5. So double-sex male gene is going to be expressed, and when we splice this down, we're going to put all these pieces together, but we're not going to have an exon 5. Double-sex female, we're going to put all these pieces together, and we are going to have an exon 5, and so that is going to make a different gene. The little black shaded bit here shows you which part of the gene actually codes for the proteins. That's the coding DNA sequence, the CDS up here. The untranslated region is the part that is not coding for a protein. It's the bit before and after the part that codes for the protein. And so you can see the double-sex female gene is actually going to make a smaller coding region, and it's going to stop here. And the male region is going to have this extra part here, and it's going to lack this little part right here. So that is how the double-sex genes, once they are expressed, are able to then go out and activate other genes. The double-sex male gene has a part that the female doesn't have. The double-sex female gene has a part that the male doesn't have, and it's missing the part that the male does have. So, all right. Now here's the sort of tricky part, but maybe not that bad. So in exon 5, remember exon 5 is the part that is different. If you had a male chromosome, you could put whatever you wanted in the region that would line up with exon 5, because you're not going to express this region anyway. You're going to skip over it when you are splicing that RNA. And so from the standpoint of having a functional double-sex male gene, you can put whatever you want right here, and it's not going to affect the actual double-sex male gene. So you can actually just make it, and you will be able to make nice little male mosquitoes. But if you put a gene drive right there, the gene drive will line up with exon 5 of the female. And if you put in the right guide RNA that says cut the double-sex female X chromosome, you will then cut and destroy exon 5. And if you destroy exon 5, you just won't make any double-sex female gene. So the AG is Anopheles gambii. That's the name of a mosquito that carries malaria. And the DSX is the double sex, and the M is for male, and the F is for female. So, yeah, that's what we've got here. And so I think you're starting to see now, maybe, how this could be used to drive mosquitoes to extinction. What you do is you put this gene, like a little Trojan horse, into the male version of the double-sex gene. And it's the double-sex male gene, DSXM. And you can see here, this sequence is actually the same sequence in Anopheles gambii and all these other Anopheles mosquitoes. So if you made a good gene drive, you could actually employ this against a whole bunch of different mosquitoes, and you could drive them all to extinction. Ugh, yeah. The way they could be driven to extinction is that if you have a male, it's going to turn off the X chromosome, and that's fine. It's still got male genes. It's got enough to live. But if you have a female, you're going to... actually, no, it's the opposite. Sorry. So males are going to assassinate their own X chromosome. And since males cannot survive without their X chromosome, they're going to die. Yeah. And that's bad. And so if you have a population that is only females and there are no males, then can you imagine what that does to the genetic structure? Yeah, you're just going to drive the species to extinction. All right. And so the way they did this was they made, look at that, there's a little homology region. So this is equivalent to the region before the break they want to make. Here's the region after the break. And they put in, at first, just a little green fluorescent protein to see if they could do that. And they checked it out, and sure enough, they were able to put green fluorescent protein in there with a CRISPR and Cas system being delivered from the other side. Now this is the really, I don't know, kind of twisted, mean, if you like, part of this. So there are differences between male and female mosquitoes. In the male mosquitoes, what they have are these objects called a clasper at the back. And so the clasper is a little chitinous pointed barbed hook. And when the male is mating with a female, you see the female does not have anything like that here. The male is actually going to slot that in under some of the plates. He's basically going to stab the female in a way that probably hurts in order to mate. And so they'll be locked together while the male is inseminating the female. The other thing that makes a male different, if you can spot it up here, look at those big bushy antennae. The antennae on the front end are really neat. They are very broad and branchy so that the male can pick up female hormones. So mating in the animal world out in nature is quite often a lot more adversarial and a lot less consent-based than you or I might be used to. Yeah, because like I tell you before, nature is terrible. Yeah, really, really terrible. And so, yeah, this is how it works. And so you can see the female is a little bit larger because she's going to be full of eggs eventually. But also she's going to be lacking those claspers on the back because she does not need to hook onto anything or anybody. And the female's just going to have ordinary antenna at the front, not those big bushy antenna. All right. So if the double-sex female gene is intact in the males or females, then what you're going to get is this. The males will look male if they have an X and a Y, and the females will look female if they have an X and an X. There you go. If the double-sex female gene is, if one copy is knocked out but the other copy is still there and it's fine, or not knocked out but genetically modified with a CRISPR-Cas9 gene drive inserted into exon 5, what you'll get will be essentially genetically normal males and genetically normal females, but they're carrying one copy of the gene. And so these males and females can mate even though they have one copy of the gene, but when they do mate, their offspring are going to get copies of the gene as well. And then the knockout version, the minus minus, is going to look like this. Because we're modifying the double-sex female gene, it's actually not going to affect the male. So you're going to get a genetically normal-looking male. He's messing up his X chromosome, but he doesn't use the double-sex female gene on his X chromosome, so it doesn't matter. It doesn't show up. But if both copies of the double-sex female gene are modified on the female chromosome, look what happens to her. Look back there, or look up here at the front end. Look at those antennae. See, that's normal female-looking antenna. Look, they're branchy and bushy. Because she is not able to express the double-sex female genes that would prevent that from happening. So yeah, that's a thing that you prevent genetically. And at the back end, look at our poor female. She is born with claspers. And the really weird thing is the claspers end up upside down. So they're going to point up. Whereas the claspers for the male are going to be down like this. The male would normally end up on top of the female and kind of locked into the back end while he's fertilizing the female. So the female is actually going to have claspers that not only is she... so a lot of the inside parts of the female are also wrong. They would be sterile even if they did not have these external traits. But yeah. So this is... a way to control the population of mosquitoes by basically, turning them to an intersex condition, somewhere in between the normal female phenotype and the normal male phenotype. And you can see it is a very misogynistic kind of thing, because we're only getting effects on the females. The males are essentially unchanged. So yeah, that's the way this particular modification works. And here's how it spreads. So first I wanted to look and see what is the viability of offspring. How many little babies are they able to have if they're carrying copies of the gene? So wild type, male and female are going to have roughly, looks like about 130 little babies. There we go, 129.5 on average, plus or minus 6. So that's why, this is why you get bitten when you go outside. Because, yeah, two mosquitoes that love each other very much can make a whole lot of other mosquitoes that love your blood very much. Yeah, and so that's where we go. And in these these mosquitoes, it is only the female that is going to need to feed on blood. So if you knock out the females, you are knocking out the itchy bites, and you're knocking out all the disease transmission that goes with the itchy bites. So if you look at this, females or males, so here's our male and here's our female, that have one copy of the mutated chromosome only, actually they have a normal number of babies, but they're going to be passing the trait on to the babies. And then in the next generation, where they're going to be homozygous for the double-sex female knockout, which is just knocking out exon 5, which is not used in the double-sex male gene, but is used in the double-sex female gene. So yeah, that's what it is. You can see that the males will have nearly as many babies. So they are still fully able to make babies with females. But since they are since they have two mutated copies, whatever chromosome they give their babies, male or female, they are going to be carrying the gene drive. And the females have exactly 0 babies. So they have claspers that point the wrong way, and they have, just a messed up uterus and stuff on the inside. And so, yeah, they do not even have a chance to be able to pass on their genes. And so once you've made genetic males that have the trait, they will spread it to females. And eventually, in just a few generations, you will end up with what I think they call on the internet a sausage party. You will essentially have all male mosquitoes, and they will run out of female mosquitoes as the females become genetically modified in each generation. And the genetically modified females can mate, they can't mate, rather, and they have 0 offspring. Yeah, so right down there. And so that's how you end up with a potentially an extinction type event. And this has been done not just in mosquitoes, it's also been done in mice. In a laboratory so far, there have been people talking for several years. You can see newspaper articles and websites going back to 2017. People talking about just releasing this in rabbits in Australia, which are invasive and fairly damaging, but wow, aiming for genetic extinction, that's pretty harsh. And once you start a thing like this out there in the world, there's no, you can't rewind the clock, you can't roll it back. If the gene is out there and rabbits are able to do what they do, or mice are able to do what they do, they will mate, they will spread the gene drive. And if the gene drive prevents like females from developing normally, for example, then you will end up with a world with no females, and that species will go extinct. So yeah, the thing with the gene drive, it's going to get turned on. The gene drives for the mosquito and for the mouse are both turned on only when the animal is making either sperm or eggs. And so at that stage, it's going to be activated. We will make the Cas9 gene, we will make the guide RNA. It's going to cut the other chromosome, and then the cell will repair that chromosome, and then it will split off one of these into each sperm cell or egg cell that it's dividing to make. That's the idea. And so yeah, within a few generations, you can end up with one of these things just traveling through the population. So modifying the double sex female gene, or modifying exon 5 rather in the double sex gene in a way that interferes with females but not males is a very good strategy for reducing the number of baby mosquitoes in the world, just like it says there. But yeah, is it ethical? Yeah, I don't know. I do not feel equipped to answer that question. We're just going through, just the facts, ma'am, just the facts there. Yeah. So the way that you make the gene drive, you're going to put in a piece of DNA that contains the following: a working copy of a Cas9 gene, because that's what's going to cut the DNA. And it's going to make a Cas9 protein. You're going to put the Cas9 under the control of a promoter that's only activated during meiosis. And meiosis, you'll remember, is when you split. It's cell division that's going to make gametes. It's going to make either sperm or egg cells. And then you're going to have next to that Cas9 gene a working guide RNA gene that will make a guide RNA specific to exon 5 of the double sex female gene. And so this thing is going to make a working machine with a guide RNA and a Cas9 that will cut any unmodified exon 5 of the double sex gene that they run into. And then either side of it, you're going to have flanking regions that look like the region just before and just after exon 5 of the double sex female gene. So you're going to cut it, and then once you've cut it, you are going to repair that gene by putting in the gene drive. And that's what they made. These are the little parts up here that they use to assemble it. The little ones with arrows are promoters. And then there we've got our Cas9 gene. And here we've got our guide RNA gene. And they decided to put it in a red fluorescent protein just so it was easier to tell. You didn't have to do a genetic test to tell whether or not they were genetically modified. They would fluoresce red under ultraviolet light. So that was nice and easy. And yeah. At first, it's going to work like a regular CRISPR-Cas9 system, but as you go on, so we're going to put one copy into the male, genetically modify them. They're going to mate with the female, and if the modified chromosome goes over to the female, great. Then she's going to have one modified and one unmodified chromosome, and when she goes to make gametes, egg cells, then all the ones with a modified chromosome. The modified chromosome is going to cut the unmodified one, break it, repair it with the modification, and bam, you're going to have two chromosomes that now have the gene drive in them. And so she's going to split those out to make eggs, and so either egg is going to have the gene drive. And it doesn't matter at that point whether she mates with a male that has a modification or unmodified, because the babies are already carrying, like a ticking time bomb, the genetic mutation that is going to drive their species to extinction. And so over time, here's what happened in some experiments. The red and blue are 2 times where they ran the experiment. The black line is kind of an average between those two, and they simulated in a computer what if they'd done this 10 more times or something like that. And yeah, over the course of 8 generations up to 12 generations at maximum, that's when they were able to drive the population of several hundred caged mosquitoes extinct. And this was done in a laboratory, so this is a controlled situation. But yeah, that's how fast the gene was able to spread. The gene drive was able to spread throughout the population. So yeah, it's kind of neat, right? Yeah. So, all right. That's a lot for you to think of, definitely. I've put a nice link to a video that talks about gene drives and gives you another perspective on that and how they work in the Blackboard. And we will be going over this again in class to try and fill in any gaps. I broke up the CRISPR over 2 different sessions because I think it's going to take 2 sessions for us to get through all of this. This is something where you got to kind of drum it in on the first session and then use what you've drummed in on the second session. So that's CRISPR-Cas9 based gene drives. Hope you found that interesting, and go out and tell other people, because you are biologists. Once you understand this stuff, you become a biologist, and there's a lot of power there, and you gotta, with great power comes great responsibility, right, Spider-Man? Of course. So use it wisely. That's all I'm saying. Or maybe don't use it. I don't know. But at least know what it is for my test. Thanks very much. That's the end of the lecture.